So good afternoon, everyone. Today we have the pleasure to receive here Anna Lapalla. I will introduce her very briefly so we can hear more and more. Okay, Anna, we are a very interdisciplinary PhD program. We have here physicists, we have mathematicians, and we have engineers and chemistry. So now I can introduce you. We are very informal, okay? So let me try to do it informally, okay?
Mm-hmm.
Anna studied natural science at University of Edinburgh. Please correct me at any point.
Absolutely, yeah.
She received her PhD in physics from Cambridge, okay, where she worked in the very famous Cavendish Laboratory. Then she worked at the chemistry department of Cambridge, okay, and then she go to Los Alamos, right?
That's correct, yes.
She worked in the Center of Nonlinear Studies, if I'm not mistaken. Then now she's an instructor at Harvard University at the Molecular Biology Department. She's actually very young. She's got her PhD, I think, in 2015, 2016, right?
Yeah, that's correct, yes. Mm-hmm.
She is a polymer physicist and now is studying glass. Now, according to she's using molecular dynamics to study glass systems. She actually gave a very nice talk at the EPS March meeting this year. So congratulations, it's very nice.
Thank you.
But her work in atomistic modeling, using molecular dynamics to simulate these biological systems, are getting very much attention at the media. You can actually see her at YouTube. That's some nice videos there. And she also very active in promoting the role of women in science. She gave a lot of talks in high schools and summer camps. So it's very nice to have you here today, Anna. I will talk a little bit more. She has we share a love for dogs. She has a very charming dog, a child named Matsi, and she is actually a very gifted painter.
Thank you.
You can see at the video some samples of their nice paintings.
Actually, I painted that last night when I was preparing my talk. So I was kind of thinking about what to talk about and how to, how to put it all together. So this was kind of a result, I suppose.
So Anna, we are here to hear you.
Well, thank you so much, Petrus and Andre. So let me just try and see if I can share my screen. This is one of the first times actually that I'm using Google Meet, so please bear with me if there's anything wrong. So, okay, I think I should be able to. I will just go into full screen mode, but let me know if you can't see properly.
Yes, we can see.
Oh, you can see.
Okay.
Can you see the full screen mode as well?
Yes, that's perfect.
Okay, thank you so much. So again, thank you so much, Petrus, for the invitation. And unfortunately, this is over Zoom, like I said, but I would love to see Brazil, and I've heard a lot about it, and it's a real honor to be here today. So I will be talking about some of the recent work that we've been doing here at Harvard, and also in collaboration with Los Alamos National Labs with Carissa Sanbonmatsu specifically and her team. And I will talk about 4D chromosome organization. So by 4D, of course, I mean, 3D in time. And you will understand why this concept of time is actually a very important aspect of our work in this particular system. And, you know, what I do is I combine polymer physics models and some concepts from knot theory and high performance computing. And that is something that I will be talking about today. And please feel free to, you know, interrupt me at any point if you have any questions, or I may take questions afterwards. So it really is up to you. Okay, so today I will start with, you know, just introducing polymer models and talk about how we can model a chromosome with our new method called 4D high C. And I'll explain what that means in a second. And then I will go on to describe a different method that's called multi-sim. And that's an approach that uses a more sophisticated way to analyze the same structures of chromosomes, but it adds a better resolution to our models. And then we also look at high experimental models that have high resolutions, and how that can also be modeled in this using this 4D high C method. And of course, I'll go into the analysis of these models and how we study the evolution of the patterns that we see in 3D. And we also will talk about chromosomes as social networks. And this is kind of somewhat a new concept, where you think of the chromosome as something that contains information, and these nodes that come together in 3D. It's almost like, you know, when people are building models about how the virus is being transmitted through a population, you can also think of the chromosome as the social network whereby certain chemical signals, for example, can be transmitted throughout the 3D structure. And so we use these social networks to track how this information is how these interactions take place. And I will briefly touch on some ideas from knot theory, where we use differential geometry and topology to understand how prime knots interact, or rather, you know, the dynamics of prime knots, really. I will not have too much on this because I thought that the main topic should be the chromosome work. But if you're interested in, you know, any further discussion, I'm more than happy to talk about this topic as well later, or by email. So let's start with the very beginnings. You know, in around, I would say, you know, 1950s, there were experimentalists who looked at the nucleus of a cell, of a eukaryotic cell, and they noticed that if you stain the cells in a certain way, then you will see these speckles. And they saw that, you know, some of the speckles have a slightly brighter color, and then some of them are a little bit darker. And so they decided to call it euchromatin and heterochromatin, just due to the different coloring. And it was later that they discovered that the heterochromatin is a very dense structure, and as a result, of course, you see that it's shown darker in this particular stain. And then in the euchromatin, you have a less dense structure, which, you know, they realized that could perhaps be approximated as an open polymer coil. And so then people started thinking, you know, is there anything more than we can understand from this? And over the years, in fact, you know, with the discovery of DNA, everything became more and more clear. So for example, here we're beginning to look at just the DNA structure on the left-hand side. And then if you add certain proteins to the DNA starts wrapping around it in a very specific way. So there's a number of tans that's very well defined around the protein that makes this compaction, this first level of compaction. And so you can imagine these spools coming together into a higher order structure. So then, you know, if you look experimentally at isolated nuclei and look at them under the microscope, so say cryo-EM, for example, you will actually see this interesting structure that's really regular. And so people started thinking, you know, what is it that's going on here? And so they termed this a 30 nanometer fiber structure because that's just the diameter of this fiber that forms. And so there's a number of models that approximate how these proteins and the DNA fold within the 30 nanometer fiber. It's still a very open question. And there is a number of models that describe this, and even studies that show that there's no such thing as 30 nanometer fiber in living cells. So as you realize, you know, in biology, most questions tend to be open-ended. And so this is one of them. If you add more scaffold proteins to the chromosome, you will start seeing even higher order organization, and even more proteins will lead to compaction in the metaphase of the cell cycle, and that is what will show you the kind of the characteristic X shape of the chromosome that, you know, most people unfamiliar with the field are used to. And so there's a lot involved, and there are many ways of looking at this from the modeling perspective. And so something that one can easily do nowadays is to directly look at the crystal structures of these individual units called nucleosomes. So this is DNA in blue, or that kind of twisted ribbon here. And those golden twists, these little coils, are actually the protein that's holding everything together, and that the DNA wraps around with. And so here, for example, you see some protein tails that also tend to be important. So another fact about biology is that everything counts. Every single interaction can be detrimental to the cell, to the structure, and to, you know, the evolution of dynamics over time. So these simulations try to take every single atom into account. And here I'm showing one of the structures that that we did when I worked at Los Alamos, and this is a structure that contains over 1 billion atoms and 427 of these spool proteins. And the structure is determined experimentally. And we use that experimental information to then take the crystal structures and make them into a fully formed structure of a gene, an individual gene. So this actually is a single gene that's encoded in this 3D structure. And even though, you know, it looks cool, there's a lot that that is still not understood about this. And one of the questions that we often get asked is, you know, is it true that this is exactly how this gene will be positioned in 3D? And the answer to that question is, we don't know. This is one of the possible structures that we can determine from experiments that give us 2D information. But realizing that 2D information can be translated into 3D in so many different ways. There's no one-to-one correspondence between 2D and 3D. So as a result, you can have a number of configurations, and this leads to a lot of quite hard questions, really. So then, if you kind of zoom out from the gene, now you're looking at an entire chromosome that here we just package this globule in a way that everything is very densely packed, and this is just, in fact, this is not even a structure of a chromosome. This is just an equilibrium coil. So this is just a polymer chain where you see the blue beads representing the monomers, and then the bonds that are between individual monomers that connect the whole system, they're shown in white. So let me briefly describe the experiments that are done to understand chromosome structure. So these were absolutely beautiful experiments. I was an undergraduate at the time, and when I saw this science paper, I was so fascinated by this that it was 2009 when I started thinking about modeling chromosomes. And the easy steps that you take really describe this work really well. So you have to cross-link DNA with a certain chemical that will put pieces of DNA together that are in close proximity to each other. Then what you do is you use a restriction enzyme that just cuts the DNA, and then you have these pieces of DNA floating around, and you mark these pieces with a protein, and then you kind of fold everything together into this kind of circle. And then what you do is you purify your DNA, and then you just sequence it. And so that result of sequencing will and these high-C of these high-C experiments will then tell you the connectivity profile for all the chromosomes in the genome. Because as you remember, the first step is to cross-link all the DNA that you have with formaldehyde, so that will create these very strong bonds of regions that are close to each other in space. And so when you look at this connectivity matrix now, you will see that red points actually represent the self-contacts on the main diagonal. So these are just the self-contacts that the chromosome has, and then everything that's off main diagonal is actually what's interesting. And of course, this is symmetrical because this is the way the data is generated. But so you can look at this triangle and notice that there are certain regions that are more densely populated with contacts, and there are some that don't have as many contacts. And then different chromosomes have different types of connectivity profiles, and then different enzymes also produce different connectivity profiles. So you have to be very careful about what you're planning to do in your experimental setup. So how do we actually model the chromosomes and the polymer in general? So here's the basic model that most people use, that I started using as a PhD student many years ago. And so the first step is to set a certain structure. And usually the way it's done is you have a self-avoiding random walk that allows you to build a structure that is not crossing itself. And this is a very vital step, because you really don't want to have knots in your structure and any problems that may result from that. So then you have interactions, the force field that you build to start the molecular dynamics simulation. And this is a 3 fold process. So first you have the non-connected beads. So the blue ones, for example, here and here, the ones that are not connected, they will simply repel each other. And so this is just the Lennard-Jones interaction that repels at a very short distance, and then you can make it either stop there, or you can have a simulation where your interaction will become attractive at a longer distance. So you can program that into your force field, depending on the profile of the chromosome, for example, or the polymer that you're interested in. And so then the consecutive beads are actually connected by a spring. So this can be a harmonic spring. We use a slightly modified harmonic spring, but the idea is still the same, that you have a minimum, and those beads fluctuate around that value. And then, of course, you can also regulate the stiffness of the chain. We know the persistence lengths of DNA, so you can in fact use that information to then build to regulate the stiffness. And in our models, this is something that's uniform throughout the whole structure. But in reality, of course, we have to take that with a grain of salt, because most chromosomes, depending on the region that you're looking at, they may be either stiffer or softer. So this kind of makes it a little bit more difficult, but nonetheless, we're just really approximating what's going on. So then you may remember the connectivity profiles that I just described, and the way we approach that problem is that during the first simulation step, you design these virtual high-C crosslinks, and so you create those directly from data. So you're not using any kind of fudging, you're just looking at the data and telling the simulation to create the connections where they need to be. So as an example, you can see here that the red dots, the red spheres, will be connected. The pink spheres will also be connected, and the green spheres we want to be connected. The rest of the little spheres, they don't have any specific interactions, so we don't care about them for the time being. So then once you run that step, once you've done that, you see that in fact this is what happens. So you force these red particles to be together, and then the pink ones and the green ones. Of course, what you can also realize from this is that this is not a single structure. So there are many states that this one high-C map can represent. So that high-C matrix of connectivity that I described, there is almost an infinite number of positions that can be found in 3D space to correspond to the same map. So here I'm just showing a brief kind of simulation result of this. So this is just a couple of particles, but you can see that there's a number of configurations that are still conforming to the rule that we give it. So the red particles are still close together, the pink ones are still close together, and the green ones are also. But of course, the 3D organization is different. So now when we look at the folding of a chromosome, things get a little bit more complicated. So in fact, here we have 833 beads for the whole chromosome. And if you look, this is the initial random walk that we've generated. And after that, you start folding the chromosome, and here all the contacts kind of tend to go where they need to be. And then you have some regions that are not defined experimentally, and those regions will actually stay the same because there's no specific interactions for those regions for several experimental reasons that I don't want to go into. But this is something that we are not really interested in. We are interested just in the structure that we get as a result of this model. So this is the final state that we're looking at now. And now we can actually also look at the connectivity profile of the same chromosome. So here you're looking at the chromosome with the different ends of the chromosome painted in pink and white. And here is the evolution of the contact map as a function. I'm not sure why this is showing as green to me. Okay, maybe now it's better. Okay, so over time, the connectivity profile changes from, you know, not having any contacts because you just have a self-avoiding random walk, to having specific contacts that are dictated by the experimental map that we just looked at. So this is the kind of the overview of the whole pipeline. You have the random self-avoiding random walk, you take experimental high C maps and feed that into your simulation structure, and you then convert that high C data to constraints. So these are going to be harmonic constraints for all these individual crosslinks, as we call them. So then you just run, once these crosslinks are done, what you then see is, you know, you can just run a Brownian dynamics simulation to generate an actual equilibrated 3D model. And then, of course, you have the computed profile that you can get from the 3D model, and you can then compare it with the original model that you have. And our comparisons show that our models are around 90% similar. So there's a lot of similarity between the actual structure and the structure that we model. So that gave us a lot of hope. Now, you know, we have a lot of biological data. The reason for doing these simulations is not simply to go and say, well, you know, this is the structure. No, we actually want to understand where the different biological things happen. And so here we're looking at two different types of compartments, A compartments and B compartments. In A compartments, shown in red, so this is all derived from experimental data, by the way. So these are principal component analyses of the information that you get experimentally. And so anything that has a positive value for the first principal component will be called an A compartment. And what people have noticed doing these experiments is that A compartments tend to have more genes that are active. So these will be the genes that are transcribed and actually made into something useful. Whereas B compartments tend to be genes that are silenced, that don't want to do anything. They just kind of hang out there and take up space. But the idea is that we can now, using the 3D models, understand where exactly these different compartments are located. And so here we're looking at the chromosome again in a slightly different configuration, and you can see that one end is very well separated from the other end. So if you just color it by, you know, the segment, you will see that the beginning is not really well mixed with the pink kind of end of the chromosome. And this is very interesting and kind of counterintuitive, really, because if you are a polymer physicist, you know that over time things equilibrate, they mix. And so, you know, if you let a regular homopolymer sit there in a folded configuration, it will just ergodically mix, and eventually you won't even know what the real structure is, because it's just mixing over time. Here you're trapped in a state where the ends are not mixing at all. So this is relevant to biology as well, because it's important to understand that everything in biology has a reason. It's not there just kind of for no reason at all. And so now you're looking at this here, and you suddenly start noticing that mostly the genes that are not active are actually kind of inside the chromosome. And here I should mention that we're looking at the X chromosome, and this is the so the X chromosome in females has two states. It can be either active or it can be shut down and inactive. And so since females have two X chromosomes, we want to compensate. So the evolution is smart enough to know that there needs to be some kind of compensation between males and females. And what happens is that one of the X chromosomes spontaneously shuts off. And so what happens then is that we can track this whole progress from the active state of the chromosome to the inactive state of the chromosome, because in the beginning you have two X chromosomes that are fully active, and then suddenly, spontaneously, one of them just shuts down. It gets covered with RNA, it gets covered with proteins, and there's not much gene expression that comes out of that. And so here you can see that the two different chromosomes now, this is the active state on the left-hand side, and the other one is the inactive state. And you can see that the structure is slightly different, but mostly what's interesting is that you see that the organization of these different types of genes has completely switched now. And it's no longer, you know, A and B compartments kind of almost face separating. You know, here you have almost a full mix of these compartments. So then what we also decided to do, you know, we felt like the 3D models that we had were interesting, but what if we decide to do more statistical analyses on these models? And so we now generate one structure from a number of structures. So we have the initial structures that are created using the self-avoiding random walk, but they're not using all the constraints. Each individual structure uses only a subset of the constraints. And so these bonds are then formed, but these different bonds form in different structures. And then you take an average of all these different states. And as a result, you, sorry about that. As a result, you get a much more statistically significant kind of comparison with the experimental data. So this is a very kind of cool thing to do. If you even average 10 simulations, 10 different structures, you will actually get a really nice, near 100% similarity between experiment and the model structure. And so here we're just kind of looking at some of the target contacts and the different contact maps that we're showing here. So these are the upper corner, the upper triangle is the empirical data, and then the lower triangle is the simulated data. And so with the regular modeling strategy, we kind of get a very fuzzy contact map, whereas with the multi-sim approach, we actually generate an almost identical structure. So here is just, you know, when we're looking at the percentage of target contacts that need to be there in the structure, we see that there's quite a significant jump between the regular procedure and the multi-sim procedure. So, you know, we then decided to look at higher resolution models and see how does this, you know, how does this scale? And in fact, we were able to increase the resolution of the data 15-fold. So now all of this is limited by experimental resolution. So you have these high-C experiments mostly are when everything started, it was all done at 200 kilobase resolution. So one of the beats would represent 200 base pairs, and that's quite a lot, and that's quite coarse-grained if you think of that as a model. But now we know that we can go higher than that and increase that by 15-fold, and so eventually we made a structure that you can see here, and this is now 5 kb resolution. And this is chromosome 20, just randomly, chosen to represent the structure. But the idea here is that, you again have the same ends of the chromosome colored in pink and white. And then as you collapse it, you can clearly see that things first are kind of still phase partitioned, but then everything starts to mix. And so this is interesting because then we can study why is it that chromosome 20 is actually much more mixed than the X chromosome. And from this data, again, we can experimentally define certain patterns of gene expression by looking at certain chemical marks. And this is methylation profile for a specific for that specific chromosome. And so this H3K27 trimethylation and this particular protein called RCOR1, you can co-localize these two. And so the red little dots are the methylation profiles and the methylation marks. And then if you look at the beads in blue, that's the protein. So we decided then to first visually inspect this, but then of course, and you can also do analysis of different types of these methylation marks, and we can see that by eye it's very difficult to see whether these distributions match or not. So then we decided that of course we can do partial radial distribution profiles for all of these, and since we have almost a spherical structure, this is kind of a reasonable approach. So then we noticed that in a specific certain methylation marks are more, they're closer to the protein, the RCOR1 protein, that the other set of methylation marks. And so as it happens, the curve in green represents H3K4 methylation, trimethylation, and this is a signature of active genes. So it just happens that this protein RCOR1 tends to mostly be co-localized with more active genes. And then so if you look at kind of the radial distribution, you can see that eventually, of course, all the interactions fade out, but with the red marks, and this is the inactive kind of gene marks, an inactive methylation profile, you can see that this is further away from the RCOR1 protein. Now, of course, there's a lot more you can do. So you can look at the volume of the of your chromosome, and now since we have a time-resolved we have time-resolved structures, we can look at these chromosomes at different points in time when the experiments were performed. So now we can look at the volume profile of a chromosome and say whether or not it changes as the chromosome becomes inactive. So we go from the active state on day 1 here in A, and then we go and fold quite dramatically on day 7. We have reached full compaction at this point. And of course, the surface area of the chromosome also decreases. Now, again, you can look at different types of genes. And what we noticed is that there are certain genes that are called escape genes. And the reason they're called that is because on the inactive X chromosomes, these genes tend to escape that inactivation of the gene. So no one really knew how this happens, but we were able to see that mostly these escape genes actually tend to gravitate towards the surface of the chromosome rather than being buried inside. They go towards the surface of the chromosome, and this is what allows them to be expressed. So then we also looked at just, regular things like root mean square displacement relative to the active structure. And we noticed that, in fact, a lot of the segments of the chromosomes that were displaced and that were displacing more were found on the surface. And then there's a region where, this displacement kind of just plateaued and nothing much happened. And yet, if you look at the core of the chromosome, the density is simply so high that there's no way the chromosome can achieve much displacement at all. So this is what led us to think that a lot of what we're seeing is actually representing a kind of glassy state where you are simply trapped. So you're undergoing dynamical arrest, at least in the core of the structure. And in fact, if you are looking at a glassy polymer droplet, you will still see that there is some motion on the surface. That's very important. So the free surface is always going to be more mobile than the core, just as a result of connectivity and density. Now, if you look at relative contact probabilities in the chromosomes, what we notice is that, the contact probability changes as a function of separation. So if you were to look at this chain as fully extended and then computed that profile of contact probability, so your probability would be low if you're fully extended, so the segments towards the ends are actually not going to have a high probability of contact in a linear chain as it happens. But if you're looking at a relative contact probability of the chromosome, it actually it has, the curve kind of follows two different stages. And so you have the separation, the stage where you're kind of in close separation. So that's the segments that are close together. And then you have a different kind of probability for those segments that are separated by a longer distance. And in fact, this is also corresponding to experimental data that they were able to determine. So these profiles are very significant in that respect. So now, if you want to go into further details and talk about the glassy dynamics of chromosomes, we decided to do a very simple experiment and look at, just a liquid globule. So you would have a homopolymer chain that's fully folded. And over time, as it explores the phase space, it actually is just behaving as a liquid. So this intermediate scattering function, you can see that it decays to 0. But if you look at the simulation of a chromosome, you start noticing that it never does. Maybe over long times, very long times it will, but not at the time scale for any kind of, relevant to the biology of the process. So we can safely say that by doing our simulations, we're able to come up with this concept of dynamical arrest in chromosomes. And in fact, we now for the first time also show that the inactive X chromosome, the one that's even more densely folded than the active one, is actually more dynamically arrested than the active chromosome. And so I mentioned the social networks in the beginning. And here's a simple example of just computing a network of interactions. And here we're looking at degree centrality for the active X chromosome in A. In B, you're seeing the inactive X chromosome, and then in C, you see the randomly folded chain. And you can see that there's a significant difference in the connectivity profile. So it seems that the chromosomes have a very tight core of important contacts that need to be taken into account in terms of structure and function. However, the randomly folded chain doesn't experience that. It has a very nice density profile of contacts, and you know, of course, as you go inside the core, there's going to be more contacts because that's just logical. However, if you look at the chromosome profiles, they are significantly different. And of course, what you can do from here is identify individual segments of the chromosome that are shown in yellow here as strong contacts. And you can map that back onto your 3D profile and identify the most important interactions that are relevant for the chromosome and its function. So here I want to kind of switch gears a little bit and talk about knotting in very general terms. So here I'm just introducing Frenet-Serre equations just so that we can define curvature and torsion. So these are concepts that I will use in my next slide to look at the torsion and the curvature in a prime knot. So this is slightly, you know, different, but I will explain why this is still relevant to the chromosome work that we've been doing. So, you know, here we just look at the profile of curvature and torsion as a function of arc length. So here on the right hand side, you see the knot, and as you kind of move it, you can see these normal modes that we're looking at right now. And so these motions represent the normal modes, and the segments of the knot that are moving the most are depicted in red, and then the ones that are not moving as much are depicted in blue. And you can just map out the curvature and torsion profile for this particular knot, which is you know, which is following the set of equations that I just showed in the previous slide. And then, of course, you can look at the relationship between curvature, torsion, and also deformation energy that I'm just not mentioning here on this slide, but you can, of course, go back to from the normal mode calculations. You can actually determine the deformation energies and, you know, looking at how it changes over time, for example, or as a function of torsion and curvature. So here we're looking at torsion as a function of curvature, and this is just a very kind of a little result that brings us to this. So now you are looking at the actual chromosome, and now you can study not just the chromosome curvature that's depicted in green, but you can also see the epigenetic marks, like the methylation marks, the chemical marks that tell us whether or not you know, the gene is active, for example. And again, since this is biological data, it really has to be heavily normalized before, you know, because otherwise you will see something like this, where, you know, you can only see that, yes, there are some relationships where, you know, if you have a dip in your black curve, there is likely to be a dip in the green curve. So there's a lot of statistics. This is very preliminary still, but this is something that I think requires a lot of attention, because my understanding of the biology of the problem is that often proteins are attracted to certain regions of curvature, and that is what can potentially lead to, you know, having a certain chemical mark attached in that specific region. So it may be that a curvature plays an important role in chromosome dynamics. And so here I'm just looking at small knots, 2 different types of knots, and we try to understand their correlated motions using covariance matrices. And so, you know, the red here represents correlated motion, and then the blue anti-correlated motions. And if you remember the slide where I showed the motions of the 3-1 trefoil knot, you can kind of see that, you know, there are some regions of this particular knot that has a higher complexity that, you know, will be moving in a very coherent fashion. And then this profile changes as your knot complexity goes up. So this represents a highly complex prime knot that, you know, has a lot of motion to it, and so this profile will end up being very, very different. So of course, what you can do with this alone is that you can start thinking about generating specific knots for a specific purpose. Now, here you're looking at a knot that has electrostatics in it, and you can kind of make it go both ways. You can make it slither through itself in you know, in a number of configurations. So it can go either backwards or it can go forward. But you can imagine, of course, by finely tuning all these contacts and the electrostatics of the knot, and adding something external, some kind of external force to it, you can actually make it into a Brownian ratchet, and that would create a kind of motion in unidirectional motion, as opposed to, you know, kind of being back and forth. So of course you can do the same for chromosomes. So here is just a correlation map, or the covariance matrix for one of our chromosomes, and you can see that there are certain parts of the chromosome that really are moving together, and there are some that are not. And so, you know, from this, the take-home message would be that there's a lot of interesting dynamical features that the chromosome has, and there's a lot to be explored in terms of those dynamical features, like the arrest, for example. So this is the end of my presentation, and I would like to thank Jeannie Lee at Harvard and MGH, and Carissa Sanbonmatsu of Los Alamos National Labs, and Maha, who's also working with me on knots. He's in the physics department here at Harvard. And of course, I would like to thank the students who were doing this work. And of course, I would like to thank you for listening and for the kind invitation. So if you have any questions, please let me know. Thank you.
Thank you a lot, Tammy. Well,
thank you, Petrus.
Our people is usually a little, so they probably be afraid to make a question something, but I will. I'm very eager to say something, to ask something.
Sorry, I can't always hear you. It kind of goes in and out.
Okay. I just saying the students are a little bit shy.
Oh, okay,
They
yes,
probably be afraid to ask something.
Yeah, of course. You know, and feel free to also email me if you're a student and you're interested in, you know, in asking a question and you're too shy to ask now. I'm more than happy to follow up with you by email. So I can send my email in the chat, so you can. Just a second. I think it's. So I think this one is going to work. This one will be my private email, so that will be the easiest way to contact me. Sometimes my department email gets a block on external email, so I don't want to risk that. So it's just lapalla.anna@gmail.com if you have any specific questions.
Do you want to ask anything?
Yes, I'm very curious about a lot of things that you show, Anna, but let me start from the beginning. First of all, I love random walks. I have been working with random walks for quite some decades right now, and Monte Carlo simulation.
Right.
And back in the time, I started with Fortran 70. So what kind of language are you using nowadays for those?
Python.
Python, yeah.
Yeah, so in fact, you know, I have to say, I also started with Fortran. The first programs I wrote were in Fortran, so it was quite a while back. But now I think that the fact that Python is what mostly the younger generation uses, and also it has the most kind of, I don't know, it's much faster than everything else, it seems, and it produces really nice graphical results as well. So it's easy to see if you're doing something wrong. So yeah, we're mostly working a lot of the stuff that I showed was actually in Python. Yeah. And yeah, so the self-avoiding random walk, you know, is kind of the really the first step to generate these simulations, because otherwise, you know, you'd really suffer if you didn't have that. Now we're trying to come up with something that would be kind of even easier to deal with, because what happens now is that we generate the beads in a way that, so we put in the first bead, and then the second one will be positioned such that, you know, it's not overlapping with the first one. But eventually, when you have many, many beads, it can happen that you will have an overlap. So then as we encounter that, we go back several steps, and then we retrace, and then we kind of do the random walk, start the random walk over again. So it's kind of like this process that over time gets more and more tricky. And it takes longer and longer if you have big structures. So yeah, it's a very neat technique for sure.
Okay, so Petrus, I have another one just because I have to leave.
May I ask now?
Yes,
yeah,
go ahead.
It's just the point. Our results doesn't depend on the initial, right?
It doesn't depend on what, sorry?
Initial condition you use. You generate
initial
it
condition,
really,
yeah,
but
it does
the
not.
exact configurations don't.
Yeah, so this is a great question, you know, because this is what we thought that would happen, because the initial condition, you know, is important in a lot of different types of simulations. In fact, if you do a homopolymer study, you know, and you have a linear chain, the collapse time for that would be very different from if you start the chain from a random walk, simply because you'll have these little nucleation sites that are formed by the self-avoiding random walk that will then fold the polymer even faster. Now, in our case, this is slightly different, because we are forcing those experimental contacts onto the system, and that is the kind of the dominant force there. So it's not, it kind of overwrites the initial configuration fully. So we've tried doing these simulations, you know, in many different initial configurations, and yet the result is always very close. So, there's only a small difference in contacts. And that's because, your harmonic bonds that are being forced onto the system are just so strong that, it doesn't matter what your initial condition is.
And use some initial velocity speed distribution.
Yeah, so actually we start with just nothing at all, and then what we do is just we turn on the harmonic constraints first. So we don't even have simulation or anything. We just identify the particles that need to be close, and that as a result kind of forces them to be together. And now only after that do we start the simulation, and then the forces and the velocities come in. But the first step is very kind of just a kind of a trick to make everything into the configuration that we need experimentally.
But I would expect that temperature is an relevant factor, and I would think that temperature will enter in the speed distribution that you use. And
yeah.
so
Yeah, I agree. I think, in order to start the simulation, you still have to equilibrate the system. So that really kind of gets rid of anything that, if there's like a high velocity somewhere and it's kind of trapped, you may really, what really helps to get rid of that is just very slow equilibration and making sure that, everything is ready to go for the simulation. Yeah. So there is a set of procedures that you need to do in order to avoid any kind of like outside velocities, like outside of a certain range, for sure.
Andrea.
Yeah.
Okay, so it's back to me. So Anna, I have a question more related to the educational system than with your simulations itself. So let try me to explain this to you. I teach to engineering here in Brazil, and in the end of the day, I end up having people coming to the PhD with us. Okay, so we have a gap between the graduation and the PhD that could be at least 2 years, but can be up to 5 or even 10 years after graduation until these people arrive at the PhD.
Yeah.
So when they're studying engineering, they have brief contact with programming, computer programming, and they normally are going to be introduced to one structured language. Okay, so they don't have contact with oriented objective programming.
Yeah.
So when they arrive at the PhD and we ask them to go to Python and program and do things like that, they come back to us and said, "Oh, I can't do it," or things like that, and they demand extra courses, and it's quite difficult in a short time period just to add all of this knowledge to them. Okay,
Absolutely.
and I think that would be similar. With happen in physics nowadays in Brazil. I think that would be similar.
It's very similar. Yeah.
How is this in the United States? Because I'm trying to get people from the computer area more close to the engineering, but I have like this clashes, and I'd like very much to hear from you.
Yeah, this is a great question. Also, even though it's kind of maybe outside of my scope. I don't know about all the universities in the U.S. What I know, what's happening at Harvard. And in Los Alamos also, it's a national lab. So I think, it's fair to say that a lot of national labs behave in the same way. But the problem is that most of the programming is actually done just by students, and they're not taught to do the programming. Almost never in their curriculum is there a course on Python programming or Fortran or C++. It's just non-existent, and it's very sad. I mean, I've had students from Cambridge who were coming to me as summer students, and it was sad because a lot of the students were saying that all they do is Excel spreadsheets, and this is physics. This is not Mathematica, not MATLAB, just Excel spreadsheets. And so, you know, all the students that I've had so far, they were the ones who were extremely excited about programming. And they were the ones who just sat there on their own and taught themselves how to program in Python. And unfortunately, or fortunately, you know, it happened to me also when I was a student. I went through the same thing where, you know, I just kind of learned, first I learned C++, which was hard. And then I kind of went a little bit off into Fortran. And then I was introduced to Python, and it just kind of grew from there. But, you know, this self-education has a lot of problems in it because you have gaps, you know, where you just kind of learn in a certain way, and you have examples, you look at the examples, you make things work. But if you don't have a class on, you know, what is programming, how to structure a program, you know, it will be a very difficult bridge to, you know, it will be a very difficult gap to bridge. And yeah, so Brazil is not the only place where this is happening. And I hope that maybe eventually this will change, and that, you know, more of the programming courses will be introduced. I've noticed that actually in engineering, it's a little bit better. The engineers who I've been working with, the students, they're much more comfortable with the programming and the different types of software. You know, if you ask them to compile, you know, software from scratch, you know, they will be able to do it. Whereas with the physicists, it's a little bit harder. And there's, I think another problem that I'm seeing here is that, you know, there's so much stuff online that people use without even understanding what it means. So, for example, you know, I've had students who come to me and they say, well, you know, I calculated RMSD, you know, and I said, well, can you show me your script? And they're like, no, I just found it online. Like, this is how I did it. But again, like, without understanding what you're computing, there's no way you can learn about programming. So that also kind of, you know, dampens the progress. Yeah, it's very sad, but I hope, you know, is there, I don't know, maybe in your department in Catalão, like maybe do you have any specific courses, or are you planning to have any specific?
Well, we are now changing our curriculum, and we are creating some kind of computer physics laboratory for each basic physics. So we have Physics 101, we have the corresponding computational course. So we introduce well, how to program and numerical methods for physics.
Yeah.
But they also have a little introductory class on computing, on algorithms, and
that's fantastic.
a numerical analysis course.
Right. Yeah, this sounds fantastic. This is, I think, exactly what a lot of the computational students will need, because, you know, if you have that basic knowledge, then doing, say, you know, MPI and working with computer clusters will be much easier, you know, once you have that basic knowledge of what, you know, how the methods work. It's really, yeah, that's great that you have that.
So we have some comments and questions.
Oh yes, so let
chat.
me see.
Wellington is.
Can another works in publisher about?
Send
Oh yeah. Yeah, of course I can, Fernando, of course I can, you know, feel free to send me an email, and if there is any articles that are not free for you, then I can just send them to you as a PDF. So yeah, feel free to contact me, and I'm more than happy to send that to you. So let's see, students who have to use free time, yeah. Yeah, absolutely. Yeah, I agree. Francioli, I hope that's pronounced correctly. I completely agree with, the idea that you have to use your free time to learn to program, or that you somehow miraculously just learn how to program. I mean, it doesn't happen. When I can tell you a funny story. After I finished my PhD, I went on to the chemistry lab where I worked with Dan Frenkel, who wrote a great book on molecular dynamics. And then when I finished that project, I went to be a software engineer for 6 months, and it was difficult. The atmosphere is completely different, and as a scientist, I had no idea what I was doing. I sat there writing unit tests for every single line of C# code. And that was the job I had, just unit tests after unit tests. But in science, we don't do that. The code I write is extremely messy. It's not necessarily the most effective, but at the same time, it does the job and it's correct. But, I think that we need to get up to the same standards, of culture, of writing code effectively, and also with unit tests. Because even, I've noticed that even great software packages like LAMPS, from time to time, there is a bug somewhere that nobody just noticed. And, probably the reason for that is that, there are scientists who work on this. And sometimes you don't have the right unit tests to pass the right arguments, for example, and then things don't quite work, even though the whole thing works perfectly, and it has, all the simulations I did were in LAMPS, and it's one of the best software packages for molecular dynamics simulation specifically. And again, I had to learn it on my own, unfortunately, because there was no way that, I would have a course on that at university. So it's, I think definitely with the Petrus, with the changes that you're having, I think it really will help your department to stand out, because most departments just don't even think about it. They're really the old-fashioned kind of blackboard style, and that's unfortunately computing. You just can't do it that way,
So, more questions? André?
Oh, actually, I'm just going to say goodbye because I have to go to another class. Thank you, Anna, so much. I'm not finishing.
Thank you.
I'm just going to go back to Petrus, but it was really nice to meet you.
Yeah.
And again, feel free and invited to come to Brazil and stay with us. Okay?
Thank you so much. I really appreciate it. Thank you.
Okay. Thank you, Petrus, for the invitation. Bye-bye.
So I have 2 more questions.
Okay, sure. Absolutely.
But remember that I'm a mathematical physicist, and so I'm interested in differential equations, and I really don't understand this.
Yeah, absolutely.
Okay. Usually when I see molecular dynamic simulations, they use some kind of solvent, usually water. But you're not using it, right?
No. Yeah, so what we use is so-called Langevin dynamics. And so this is just the Newton's equations of motion with the random force. Yeah, so it's another trick that you can do. And the reason for that is simply to save the compute time. Because if you were to simulate the solvent particles, that would add, a great amount.
And you can't
it would be too much.
Yeah, the expected value of your moment and simulate some kind of temperature also.
Yeah, exactly. Yeah, so you can still, so you can change the temperatures with the Langevin thermostat. It's still all possible. But, there are, of course, limitations to the Langevin dynamics as well. The fact that you will, if you want to simulate something that involves hydrodynamics, for example, you will then have to use a different set of algorithms to achieve that. And there the problem becomes, the difference in timescales, because you have a system that you are simulating, and then you have the kind of the hydrodynamic simulation running almost on its own. And those have two different timescales. And then like combining all that information and making sense of it is sometimes tricky. So, but the simulation that you mentioned, the billion atom simulation, in fact, most of the atoms were water molecules, and it's very computationally intensive to compute all that, because it's just too many of them. And then if you think about the trade-off, between between simulating the actual system and then just simulating water, you have to pick. Yeah.
So my final question is, so use some kind of social network tools to and particularly you analyze connectivity.
Yeah.
So that's some kind of power law, or you don't?
So yeah, with the connectivity, there are some power laws in some cases. I can't remember exactly. Some of the profiles definitely had very interesting power laws involved. But there's different types, it really depends on the system. And one of the problems with biological systems in general is that there's so much noise that's associated with it that, sometimes you may see something interesting, some very interesting kind of response in one simulation, and then you run another and it's not there anymore. So, that's something that I'm really interested in, trying to see if there's any interesting relation in the connectivity profiles and like where exactly they're positioned in the 3D space, and everything related to connectivity, because that's effectively what will drive the dynamics. The fact that high densities really prevent the structure from, it's almost like jamming, a jamming transition, where really it just jams at some point, and there's nothing you can do about that. And only large fluctuations will be the ones that actually can get the system out of that jammed state. Yeah. Oh, I see that somebody raised the hand.
Domingos? Domingos is up.
Oh yeah, hello Domingos.
Hello, Anna.
Hello.
Thanks for the presentation.
Thank you for coming.
Just a curiosity. In your presentation, you show a simulation that a million of time steps, and I would like to know how long does it take, and what machine that you use for that.
Yeah. Okay. So for the smaller simulations, are actually the ones where I'm simulating the chromosome. So what I mean by time steps, or sometimes I call it tau, the time. So this is Lennard-Jones time. This is reduced time. So we're not talking about real units. We're talking about reduced units that can effectively be converted to real units if you want to do so. But so for the chromosomes, the simulations actually only took about a couple of hours, maybe. So and that was done on just on my laptop. So you can really, even with one single processor, you can really do these simulations. And that's one of the reasons why, we think it's very effective. And what we are planning to do next is to have a web server that allows you to run these simulations, because it's, with the fact that it's so quick can allow anyone, if you're, say, an experimentalist, you can just put in a connectivity profile and out comes a structure. So yeah, within a couple of hours. And of course, for the billion atom simulation, you know, of the single gene, that took 3 or 4 months of simulating it on the full Trinity cluster. I don't even remember how many cores that is, but it's hundreds of thousands. Like it was a lot. Yeah. So you know, you can really with the coarse-grained approach, you can really kind of take it to the minimal. It's almost like a minimal system where you can just work with a very you know, with very brief simulations, and it really produces a quick result, and you can see where things went wrong, for example, and how to make the simulation better. So that's one of the kind of positives of that approach.
Yeah, it's a fantastic thing to can simulation a work like that in a laptop. It's
yeah. Thank you. Thank you.
Thanks.
Thank you so much. Thank you, Domingos.
Any other question, guys? Oh, so I will thank you again, Anna.
Well, thank you so much, Petrus, again for the invitation, and thank you everyone for coming. This was such a pleasure for me to talk about this. And,
it's our pleasure.
Thank you. Thank you.
So Paulo sent congratulations in the chat.
Thank you, Paula. Thank you. Well, and like I said, please feel free to email me if there's any more questions that you can think of, or you know, if you're interested in any kind of collaboration as a student, even if you're interested in some kind of project, then, you know, I'm more than happy to think about that and maybe come up with a plan. So.
Oh, that's great. So let's thank Hannah again.
Thank you so much, everyone. Thank you.
That's great. So till next Friday, guys.